Well, thank you very much to the foundation, the Ferrero Foundation. Although I'm Spanish and I understand quite well when you speak in Italian, I think I'd rather not speak in Italian because this will turn up into a stand-up comedy. You will laugh a lot or not understand anything. So I will try to explain myself in English. The topic I'm going to talk about is very complex, multidisciplinary, and I'm really proud and honored to be here and listening to the world-class people in the area of microbiota. We are newcomers in the topic, so you will find that I simplify most concepts and ideas, including information. I'm sorry for this, but I think we have to consider all factors. I'm also going to, of course, have to I don't find we'll have to talk a little bit about cognitive impairment and topics that will be dealt with tomorrow. So I hope I'll not make a spoiler on some of the issues that are going to be discussed tomorrow. I'm not going to talk in depth about demographic problems. I'm not going to show demographic pyramids. I just want to show a real problem, which is the growth in population and the growth of the dependent elderly above 70. If we take the whole world population, you will see that elderly above 70, when we relate to other adults, give us the dependency ratio. With the whole world population, I arbitrarily draw a line on the 25% of dependency to compare each of different parts of the world. You will see that the whole world population, including all developing countries which have a lower age, will come to the 25% of dependency in about 2080. However, in the United States, in North America, these 25% of dependency will arrive in 2030, and in Europe a bit before, and in Southern Europe, which we are much long-living people, we are just about to reach this 25% of dependency, means 25% of older than 70 in relation to other adults. And we have to learn about the solutions that they have implemented in Japan, because they have reached this figure much earlier. In relation to cognitive problems and dementia, if we take global numbers, the low-income countries are going to grow much faster than the high-income countries. Still, the figures are very high. In this graph, we find the high-income countries, Europe and North America, together, and this relates the percentage the per 1,000 of dementia and cognitively impaired people per 1,000 of elderly of all of these ages. What this is meaning is that in this room, if we are approximately 100 people, those of us that are hopefully going to arrive to 80, or 85, or 90, among all of us, 10 to 15% will be dement, and maybe 20 or 30% will have severe cognitive impairment. That, personally, I'm closer to this age, not very much, but I'm getting closer. So that's why I took this project. In worldwide, if we want to convince people and the governments to finance this really means an astonishing amount of money to be spent in taking care of dementia people. Risk factors have been analyzed already for Alzheimer's disease and dementia, and they are first, for the early onset, of course, it is genetic, and it comes up in young people of about 40. Late onset sporadic Alzheimer's disease is multifactorial, and higher risk is found in with the presence of the APOE homozygote epsilon-4 allele, and also with sorting related. Amyloid beta and microfibrillar tangles, of course, are diagnostic factors that are associated with risk, but the highest, the factor with highest risk is age. But let's see a bit of light, a bit of hope. There are recently been defined a number of functions that are modifiable risk factors that have been related to dementia and cognitive impairment, and they're called modifiable because they can be controlled. Like in this article, hypertension, obesity, and depression are really related to Alzheimer's disease, to the cognitive decline. A number of organizations and foundations have made reviews and meta-analysis on these modifiable risk factors for cognitive decline and dementia are the same. With certain values, they increase the risk, but if they are controlled, they decrease the risk of cognitive decline and dementia. And they can be summarized in disease management, disease management factors like diabetes, midlife obesity, cardiovascular disease factors like hypertension, and lifestyle risk like current smoking, physical activity, diet, alcohol abuse, cognitive training, social engagement, etc., and depression and sleep. We will come back to these modifiable risk factors because, as you can already imagine through the conference that you have listened before, these factors are mostly related to microbiota, are associated to microbiota. Dementia and Alzheimer's disease are bound to inflammation. Sorry for this, it's an oversimplified scheme. This has been dealt with extensively before, so I will not come to make a lot of noise about this, but of course, older people have a reduction of immune response and a low grade of systemic inflammation. This can be measured and related as markers to cognitive impairment and Alzheimer's disease and dementia. And in a previous work that we developed with sepsis in neonates, we could put forward a model by which systemic inflammation would be stimulating local different organs, inflammatory pathways and oxidative stress pathways, and in the case of the gut, these will turn into the synthesis of antimicrobial peptides and possibly reactive oxygen and nitrogen species that have antibacterial effect. And this theory comes from experimental data from Sebastian Winter in the University of Texas that has demonstrated that particularly the reactive oxygen and nitrogen species are donors of electrons for electron transport pathways in certain bacterial groups that use them for active respiration, like enterobacteria and staphylococci. Gut microbiota in older persons has been very, very well explained by Paul O'Toole, so I will just and by Professor Franceschi. So I will just summarize that there are differences in centenarians with healthy elderly and healthy adults, but healthy older persons have a normal microbiota, a normal adult microbiota. And as he has explained, well, this is linked. Certain species are linked to the production of pro-inflammatory cytokines. Also, it was explained that very nicely that the gut microbiota in elderly was first grouped by institution and community, but in fact, if I understood correctly, this was rather due to the diet. And also they stressed that first, this work stressed that the diet is very influential on the gut microbiota, and second, that healthy, if I did understand correctly, healthy adults have a similar microbiota to healthy elderly. So gut microbiota in Alzheimer's disease and cognitive impairment. Let's see what we know. We made a very small work on transgenic mice, double transgenic mice, and we could notice that not when they are young, but when these little female mice grew, then we could differentiate the microbiota in transgenic and their healthy homozygote sisters. The difference was mainly due to the presence of abundant proteobacteria that, as has been mentioned already, proteobacteria are related to inflammation. In Alzheimer's disease, there are few works that I could find, but I see very little here from here. But you can see that ruminococci are abundant in the healthy controls, also bifidobacteria, Clostridiaceae. This family of bacteria includes some groups that have been mentioned, like Lachnospiraceae and Clostridium group 4. And in Alzheimer's disease, we found that in this paper that Bacteroideaceae are overrepresented, like Rickenellacea. This fact that Bacteroides group is highly present is quite interesting. Sorry, that was, let's skip it. In another very interesting work on Alzheimer's disease microbiome, they show that certain bacterial groups are related to the dysregulation of the anti-inflammatory of P-glycoprotein pathway, linking bacterial groups to the inflammatory profiles in Alzheimer's disease. And they also find Bacteroides increased in Alzheimer's disease, Alistipes, Odoribacter, and Bernacella. And again, Lachnoclostridium, which is the group of Clostridium and Lachnospira. And other dementia have a similar pattern, not Bacteroides. An interesting group by the group of Paul O'Toole, an interesting paper by the group of Paul O'Toole revealed the linkage between this APO-A genotype that is related to the high risk of Alzheimer's disease and Ruminococcus, to Ruminococcus and Prevotella and Roseburia. So it's all indicating that these diseases, as has been already explained, have a connection with the gut microbiota. Our very modest participation in this topic, it comes from a small study of 35 older persons where we wanted to see if there was a difference between mild cognitive impairment and normal cognition. First, of course, we didn't make a selection for age. There was a wide range of ages, and first thing we found is that the mini-mental results were linked to age. So from the first analysis, we had to discriminate and pull together by age groups. Then, interestingly, the age was bound to inflammatory markers, the myeloperoxidase, but also the results of mini-mental, and interestingly, to the antibody titers to Epstein-Barr virus, which is an herpes virus, which has been already described to be related to general inflammatory responses. Then general microbiota analysis showed that the profiles of bacteria were different in normal condition volunteers and those that had mild cognitive impairment. And the Pearson correlation showed that certain bacterial groups like Enterobacteriaceae and Christeneliaceae were significantly linked to cognitive tests and to the inflammatory markers. The dominant groups, bacterial groups, can be separated by, if we take the families, we see again that Bacteroideaceae are present in higher numbers in the normal condition group, and Akkermansia, excuse me, sorry, and the Akkermansia group, which is Verrucomicrobiacea, is present in the cognitively impaired group, like Fusobacteria. If we come to species, we find Bacteroides and Lacnospira in the normal cognition, and Ruminococcus, one Ruminococcus species, and Akkermansia monsinifila in the impaired group. Of course, we have a very small amount of volunteers, and we could do what we could do. We didn't find, because we did not select for deep cognitive impairment. Maybe this was also selecting against the homozygote epsilon 4, but we found different groups in the heterozygous epsilon 3, 4, and double epsilon 3. And again, we see familiar names like the Lacnospiraceae bound to the homozygous epsilon 3, while the risk factor epsilon 4 was associated to Christeneliaceae and Akkermansia again. These are names that are coming up again and again through this afternoon, as you will see. Then, well, we also analyzed the oral microbiota, and the results are not ready yet for publishing, but we found some interesting differences in the relation to the cognition. But the last part will be a more general observation on factors which are affecting gut microbiota composition. Of course, I will not mention, because it was obvious, the administration of antibiotics changes completely the gut microbiota, so that will be factor number 1. I did not include it. Factor number 2 is the environment, although it should be diet, but I just like this picture, is the Yanomamic tribe, and it's a very well-known study where Dominguez-Bello group demonstrated the presence of more diversity in these newly known Indians in the Amazons of Venezuela than in the American population, and also by the patterns they separate with other, let's call primitive tribes. Very interestingly, the environment has been shown to be bound to a decreased, the children grown in farms and in contact with farm animals are being demonstrated to have a lower risk of allergies and asthma, and it has been recently shown that this is being due to the microbiota. In fact, when they distributed dust on the floor of a number of houses, this can be done only in the northern Scandinavian country. I don't see this in a Mediterranean country, a mother allowing someone to spread dust all around the house for the child to crawl around and get the dust. They could show that these decrease significantly the incidence of allergies. Diet, of course, it has been already mentioned. It's just passing by this factor. The entero groups defined for a long time already in meta-heat and the posterior groups that split the profile of microbiota of populations according to the diet, whether they are Bacteroides or Probiotica, according to the intake of vegetable fiber. The Mediterranean diet, adherence to the Mediterranean diet apparently also closes the microbial profiles to the vegetarian profile. Then other factors like obesity and diabetes. For this, of course, we have to mention the O'Toole's group work, initial group on the transplantation of microbiota from obese mice, that they transfer the weight gain ability to lean mice. But there are many works, and this may be one of the health issues that has been more deeply studied. Hypertension has been recently related also to gut microbiota, like increase in Prevotella, Pasteurella, Escherichia, and Shigella, and the controls had greater Bacteroides and Faecalibacterium. Physical exercise, lifestyle factors are more difficult to relate to gut microbiota, although they can be. But physical exercise apparently does not bring a significant difference in the profile of microbiota, but in the behavior of the microbiota and the genetic content. Excuse me, I don't know what I've done. Sorry, time. Okay. Also, smoking seems to sometimes, well, in one small study, they show that giving up tobacco changes the gut microbiota, but in very low significance. A deeper study showed that oral microbiota did not change with smoking. And of course, the pioneering group in gut microbiota and gut-brain axis is the Cork group, led by Professor Krajan Dinan, and they defended that especially anxiety and depression are linked to gut microbiota. These are very preliminary, very early studies on germ-free mice that have been, of course, overruled by more recent works. And also, this last study showed that depression and mean quality of life are associated to Bacteroides. What I wanted to come is to this slide again, where we put together all these modifiable risk factors for cognitive decline and dementia, and relate to gut microbiota. Evidently, diet, obesity, diabetes, cardiovascular disease, hypertension, to lesser extent physical activity, smoking, and possibly depression and alcohol overconsumption are directly related to microbiota. Those in medical terms of risk for cognitive decline and dementia are classified as controllable factors. So gut microbiota, it could also be a controllable factor or a master factor that interconnects all of them, as it has been proposed right before me. Classical probiotics have been studied a lot, and it has been shown that they can improve the mood, that they change partially the proportion of bifidobacteria and lactobacillus in the donors, and they may improve immunological parameters. Finally, the modern techniques allow to analyze microbiota where we did not reach before, and it has been shown recently that, or it has been described that some bacteria can be found in the brain, some yeasts too. At least it has been reported. And certainly systemic inflammation may alter the blood-brain barrier and allow, in some pathological cases, the passage of antigens, as it has been shown by lipopolysaccharides and perhaps microorganisms. I just had to mention this because we are talking about brain diseases. My conclusions are that, of course, age and inflammation are main risk factors, and those are not my conclusions, are the general conclusions of people working on this topic. Main risk factors for cognitive decline, dementia, but they can be controllable factors. There are many that reduce the risk of cognitive deterioration. Competent cognitive condition correlate with the presence of Bacteroidetes in our study, bifidobacteria, Lactospiraceae, and other short-chain fatty acid producers, like those producing butyrate, acetate, and propionates. And cognitive impairment is related to an increase in Cysteinelaceae and Enterobacteriaceae. While in Alzheimer's disease, they describe Bacteroidetes as an increased group. Some probiotics can moderately modulate inflammatory markers. And of course, the main conclusion that I would like to show is that data that have been accumulated through recent work by others and modestly by our small contribution is that gut microbiota can be considered a central modifiable factor to reduce risk of cognitive decline, and maybe old but new type of probiotics, let's call them probiotics, could have a vital relevance, as it has also been proposed right before me. And that's all.